with Lexagene. Again, the purpose of today's presentation is to introduce the company and to give you a better understanding of the business through a PowerPoint review and discussion with management. That discussion will be led by CEO Jack Regan, who is also joined on the phone by President Daryl Reback. We're going to break for Q&A at the end of the presentation. When we do break, we encourage Q&A to take place. And just a reminder, we're only going to take questions via the web portal, so if you're listening over the telephone, please access that web link that I sent earlier to ask that question. Again, you'll be advancing the slides on your own today using the arrow keys on the top right-hand corner of the presentation. And you can also expand the PowerPoint using the expansion feature in the top right of the PowerPoint. Should these arrow keys disappear, simply hover your mouse over the top right portion of the PowerPoint of your screen. And if for whatever reason your audio is having any issues, a dial phone number has been emailed to you. Or again, you can submit a question using the question text box within the webinar portal at any time. I'm going to ask that question on the air for everyone to hear, and management will then answer. I'm not going to reference anybody by name, but simply read the questions asked. And if I can't get to your question online, I will come back to you via email. If for some reason you're experiencing any issues once we start, please remember you could email me also at glenn@bristolir.com, and I'll be happy to assist. I'm not going to read the forward-looking statements, but simply state that they do apply, and I reference them on page 2 of this PowerPoint. With that said, once again, thank you for joining us. Remember, this is fairly informal, and we do encourage lots of Q&A to help better understand the business and its growth path. And I'll now turn the call over to Jack to start his part of the discussion and presentation.
Okay, thank you, Glenn. Thank you for the introduction. And to the participants, I do appreciate the opportunity to present Lexagene to you. So Lexagene is a med device company. We are working on developing a fully automated genetic analyzer. We refer to it as LX2. It's shown here in the cover slide. This particular instrument is very well suited for detecting pathogens. A model pathogen is shown. It's E. coli. I'm sure you're all familiar with it. And when dealing with pathogens like E. coli, if an infection is present, you can appreciate the importance of having both an accurate and timely result. And this technology is ideally suited for that. It's designed to be placed at the point where samples are being collected. This eliminates the need to ship a sample to a reference lab, thereby saving a tremendous amount of time. And furthermore, it only takes an hour to return a result. So what makes this instrument so powerful is that it employs an automated genetic analysis. We perform a chemistry called PCR, and that effectively amplifies the target sequences a trillion-fold. This means we're extremely sensitive in making us extremely accurate and really a well-designed instrument for many different applications, which we'll go into during this talk. So with that introduction, we'll skip forward to the third slide. I'll provide some corporate highlights. So at the very highest level, Lexagene is working to provide a high-quality answer that is on par with answers provided traditionally by reference laboratories. So the way in which we're going to do that is by, of course, we've reduced this to practice now, is a microfluidic instrument that is fully automated and returns results in just 1 hour. So this company came into existence in 2016 through a reverse takeover agreement. At the time, we just had 2 patents. So since then, we've grown the company to 21 employees strong. We are headquartered just north of Boston. And during the course of the last 2.5 years, we've completed 2 financing rounds, bringing in over $13 million US. Some details on our cap tables listed below. If you're interested in learning more, I encourage you to reach out to the company. I'll get you in touch with Jeff Mitchell, the company's CFO, and he's happy to answer any questions you may have. So on to slide number 4, a little bit of history of the technology, how it came into being. I am the inventor of the technology. Back post-2001, and you may recall 2001, we had terrorist attacks on U.S. soil, both by air and also by mail. What came in by mail were letters containing anthrax that went to senators and also to certain media outlets. And as you might imagine, when you target senators, a lot of money ends up being ear tagged for technology development to better prepare the country for a bioterrorist attack. I was involved, a lead scientist on a group of talented scientists and engineers developing automated instruments to better prepare us for the next bioterrorist attack. And we designed an instrument that ended up becoming validated by the Department of Homeland Security. What that means is the Department of Homeland Security trusted the technology well enough such that if it generated a positive result, it would take decisive action, including shutting down an airport, shutting down a train station, evacuating Congress, etc. And so I described this instrument in a manuscript that's published in Analytical Chemistry. I am the lead author on that manuscript. And through my experience working on this technology, I certainly could appreciate the advantages of microfluidics, but I certainly also recognize that the technology could be modified and improved such that this biothreat detector could really be applied to many other markets. So through the course of creating a new design, and this new design really made this instrument, which was solely for biothreat, now much more powerful, able to be used in addressing markets like veterinary and human clinical diagnostics, food safety, and others which I'll go into later. We ended up filing for patent protection, and those patents, were issued. And since Lexagene has acquired exclusive rights in certain key markets to market this technology. So moving on to the next slide, slide number 5 now, key investor considerations. Again, we are really competing against a reference lab model where samples are being shipped and answers are being returned 2 to 5 days later. Instead, we are promoting a fully automated instrument placed at the point of sample collection that returns a result in just 1 hour, again, rather than waiting days. This means that the instrument must be able to take in a raw sample and pump out a genetic answer and be extremely easy to use, and we've achieved that. In order to be a good business, we have to obviously make money. We are pursuing a razor blade business model. We do have a consumable, which I'll get into in a bit. The margins for selling that consumable are projected to be extremely healthy because it's very low cost to manufacture. So since reducing our technology down to functioning prototypes, we have generated 4 data sets, and we're very proud of these data sets as it's putting a stake in the ground in regards to our capability. Again, there is a slide later in the slide deck that speaks to these data sets, so I'll go into more detail later. And as I mentioned, we have patents that have been issued and licensed from Lawrence Livermore National Labs. And Lexagene itself has filed a patent which is pending, and we have other patents which are currently in the works, which we will press release upon at the appropriate time. So with this technology, we are going after what we consider to be underserved markets. When I say underserved, these are markets that currently don't have access to high-quality automated genetic testing instruments. Collectively, they're valued at over $40 billion. Our primary market right now is veterinary diagnostics. We'll also get into food safety, as well as open access use, which I will describe in more detail in a coming slide. Slide number 6. This is, again, a closer viewpoint of our LX2 instrument, which we anticipate commercializing. I would like to walk you through how you would interface with this instrument. It has a touchscreen monitor. There's no keyboard involved, so someone interested in running a sample would collect a sample. They would load a sample onto the face of this instrument, and at the face of the instrument you'll see a couple key features. There is a vertical slot which contains enough room to place a vacuum container, that is a vial containing liquid. Above that is a horizontal slot which is designed to receive our single-use disposable cartridge. And above that is a lever that is designed to be pressed down, thereby clamping our disposable cartridge to be in line with the microfluidics of the instrument. At that point, the user would use the touchscreen monitor to initiate sample processing. They walk away, come back an hour later, and they have a result. It's really that easy. There's no pipetting involved. Another key feature I want to point out with this image is down below you'll see a green and a blue cartridge. These are reagent panels that hold test material, and these test materials are specific to certain genetic sequences of interest to us, whether it be a pathogen or a marker such as an antibiotic resistance factor that we believe is important for the particular application that it's being used for. So on to the next slide, slide number 7. Our technology certainly is unique. We view it to be unlike any other on the market and have, excuse me, many competitive advantages. At this time, I'll spend a moment describing what open access means, because far and away, there is no other technology in the market that's easy to use and also open access. So what open access means is that generally performing genetic testing is a very complex process. It requires skilled molecular biologists to do the work in an equipped laboratory, and it takes a lot of just expertise. What open access means is we want to give people who are doing that genetic analysis the ability to do it automated, but furthermore, the ability for them to customize their test to screen for whatever they want to. This is the term open access. When you have the ability, or granting the end user the ability to use an automated instrument, and they can dictate what the instrument should look for. In contrast, other automated instruments on the market today are closed access, they're fixed, they cannot be changed. This ultimately blocks many potential customers from buying their technology. As I indicated in the previous slide, ease of use, really touchscreen monitor, no pipetting, non-technical people are intended to use this instrument. Really less than a minute to initiate sample processing. Low cost, we'll get into this later on. Our cartridge and reagents, the way they're manufactured, extremely low cost, allowing us to outcompete competitors in regards to cost of testing. This also allows us to access markets which are very, very cost sensitive. Lower right-hand corner, I do want to mention the differences between genetic analysis and culture. Not all testing is equivalent. Reference labs often rely on culture. Culture is the process of taking a sample, placing it into an enriched broth, placing that mixture into a controlled environment that's heated, waiting generally overnight, and then following that with additional analysis to figure out what was in the sample to start. It's a very lengthy process, and it also has some drawbacks. First, in regards to time, time has consequences, also has a cost factor associated with it. Again, to contrast it with what we're doing, we are avoiding shipping and placing an instrument at the point of sample collection, thereby entirely removing the shipping time. Likewise, we don't have to rely on culturing the potential pathogens in the sample, another tremendous time saver. Genetics allows for extreme sensitivity and specificity. I mentioned early in the cover slide portion of the talk that we are automating a process called PCR. PCR is an exponential process. It results in a trillion-fold amplification of the target sequence. This means that if the pathogen is present, we certainly should detect it. This is a tremendous advantage, and it doesn't make a difference as to whether or not the pathogen is live or dead, and this also has advantages in regards to improving your sensitivity and identifying samples that may be of higher risk. Also, genetics allows you to largely avoid some false positives and false negatives if your assays are designed appropriately. When you are shipping samples to a reference lab, that shipping actually is a risk point where either you can get unintended growth causing a false positive, or you can actually kill the organism you're hoping to culture eventually, thereby resulting in a false negative. By doing the testing at the point of sample collection, you avoid this risk and generally get a higher quality answer. Genetics also allows for one to test for multiple pathogens at once, and we don't care if it's bacteria, fungi, viruses. No specialized culture conditions are required. We can just test right away. Again, a huge advantage. On to slide number 8, the improved user experience. Again, in order for a technology to be successful in multiple marketplaces, first, they must be easy to use. There are many instruments out there that require very skilled people to operate them around the clock. That is not the case with our technology. We expect training to last really less than 5 minutes. An operator simply needs to collect a sample. They can load up to 2 samples at a time. I do want to mention that our technology is modular in the sense that we certainly can build our technology to process more samples at a time to really meet the demands of a particular laboratory. These samples are processed in an on-demand fashion or a stat fashion. Running throughout the day, you certainly could process roughly 48 samples within a 24-hour period. The instrument does take in a liquid sample. It could be of a variety of different sample types, urine, the media from a swab, sputum, etc. Any type of a liquid sample the instrument can process. And I've already mentioned the low amount of time for initiating sample processing. Once that button hitting, like the go button, so to speak, on the instrument is initiated, the instrument takes over, and it first concentrates the pathogens in that liquid sample. It extracts and purifies the genetic material. Again, we're looking for the genomes. And it sets up and screens for about 28 different targets. And these targets can be particular pathogens. They can also be antibiotic resistance factors or other things that the end user deems to be of importance. At the end of PCR, the instrument goes through an auto-analysis. It compiles the data, and it assembles a PDF. And the instrument will email a PDF to the operator. Thereby, the operator does not need to stand by the instrument waiting for it to finish. That operator can be at their desk or effectively be monitoring their phone and get the result. I do want to mention that these are steps, again, normally done in a reference laboratory with a skilled molecular biologist using multiple pieces of equipment and refrigerators and freezers. It takes hours, and we are reducing this complexity really down to a very easy-to-use, extremely powerful microfluidic instrument. Slide number 9, and now moving to slide number 10, the addressable markets that we are targeting. Again, this is over a $40 billion market opportunity. We view veterinary diagnostics to be a greenfield opportunity. Right now in this sector, there are no point of care, if you will, automated instruments that provide a high quality answer. Therefore, veterinarians are forced to collect samples and ship them to a reference lab where they wait 2 to 5 days. That's really inexcusable at this era of microbiology, and we certainly believe we can provide an instrument to them which will solve their problems. Right now, companion animals is a booming market. Pet owners will pay handsomely to maintain the health of their dogs and cats. It's predominantly a cash business. It is a great business for us to target and get into as really first-to-market advantage. Food safety is another big market for us. It largely has been ignored by genetic testing companies because it's a cost-sensitive market. Nonetheless, because of the profit margins of our technology in regards to every sample processed, we can still provide the technology at a low enough cost and provide enough advantages for this really to take off in this sector, and again, for us to have a huge advantage really being first to market. I'm sure you're all aware of the impact the foodborne illness outbreaks have had on the industry and can appreciate that there's a lot of demand from the consumer side, from the government side, as well as from the food producers and packagers who want to make sure they're providing the safest possible product and also the freshest product possible. Down below we have clinical research and diagnostics, likewise other open access markets. There's a lot of bullet points here. I'm not going to go through them in detail. My point here is there are many, many laboratories across these industries that perform manual PCR. This is the genetic test that our instrument automates. And in doing so, there's a lot of overhead and costs with manually performing PCR. They certainly are looking for ways to automate that process, to remove the human factor, if you will, to have the reliability of an instrument. And because we are open access, that means that once we start selling our instrument, these labs that have tests in-house already, they simply need to buy our instrument, load their tests into our instrument, and they're off and running. Importantly, we are still tied into their a revenue stream from them because of the disposable cartridge, which is required every single time you run a sample. On to slide number 11, the veterinary market. What's really going to drive adoption here? Put yourself in the shoes of a veterinarian. Right now, veterinarians see sick animals, the dogs and cats. Unfortunately, because they do not yet have automated high-quality testing at the point of care, they're forced to ship samples to a reference lab and wait for a result. Meanwhile, you have a pet owner who is really pleading for some assistance and some therapy for their sick animal. And so the veterinarian goes through their process of empirically diagnosing and prescribing a therapy. What empiric means in this case is a best educated guess, because that's all they have really at their disposal. They certainly would prefer to process a sample in an automated instrument, have a genetic report that identifies the cause of the infection and also gives a guidance in how best to prescribe so that they are not under or over prescribing, but appropriately prescribing for that particular animal. So we certainly believe we'll be able to provide that solution for them. The market is sizable. You'll see there's over 25,000 pet hospitals. There's a lot of dogs and a lot of cats in the United States alone. Obviously, the worldwide numbers are substantially greater. This last bullet point, 5.4 million UTI. UTI stands for urinary tract infections. These are run every year in the United States, but I do want to point out that our instrument is not just intended in the veterinary market to address urinary tract infections. We anticipate eventually our technology will be a one-stop shop for the veterinarian in the sense that if they have an animal that's suffering from a respiratory illness, they can take a nasopharyngeal swab, place on the instrument, and we'll give them effectively a report that they can then use to diagnose the animal. Likewise, for gastrointestinal diseases, for wounds, and other such, we really expect to be able to largely replace the need and reliance on a reference lab and thereby really empowering them to better perform their job and give better managed care to their patients. Next slide, slide number 12. Food safety. In the United States every year, there's roughly 48 million foodborne illnesses. We've all known the horrid feeling of being sick with a foodborne illness and can appreciate the frustration that we're not better protected. And despite efforts, we have not been able to reduce the amount of recalls really happen on a yearly basis. As such, the government, through the Food Safety Modernization Act, is really pushing for new technology adoption. One of these is the employment of genetics to help out, and I was invited by the USDA, specifically the Food Safety Inspection Service, to come down to DC later this month and give a talk detailing how genetics can help this industry. And we certainly think we're well positioned to make a big impact. And the way in which we will do that is we'll be another tool for food safety officers to better understand the risk certain samples present so that they can ship safer and fresher products to customers. On to slide number 13, the open access. Again, as a reminder, these are the labs that currently do a lot of genetic testing. They have tests in-house, and they're desperate for automation. These are an abundant number of opportunities for us across the pharmaceutical industry, the biotech industry, universities, and it goes beyond that. This list is not comprehensive, but water quality testing, agricultural testing, often underappreciated. We often think of pathogens just affecting humans predominantly and then animals, but of course pathogens also affect plants and can be devastating to farmers. And the agricultural testing industry is a significant industry. And it extends also into biothreats. I mentioned my previous experience in the biothreat sector, and of course in pandemic prevention, where the utility of open access to help protect against the next pandemic strain is extremely important. Slide number 14, we're going to the supporting data and comparative studies section. Now on to slide number 15. So when we first reduced our technology down to a working alpha prototype, we performed a study focused on canine urinary tract infections. So we processed 107 dog urine samples, and this was right out of the gate once the technology met our specifications for performance. The results came back 97.5% concordant with reference lab data. And as I mentioned at the start of the talk, our goal is to provide reference lab quality data, but not in the time frame of days, but in the time frame of a single hour at the point of sample collection. So there is an asterisk on this point because although we're very proud of this first mark in the sand, we certainly expect to be able to improve this. One of the powerful aspects of our technology is that we can very quickly update our genetic tests to detect things that we potentially missed. Every time we perform a study, we look to see what we missed, and we look to correct that and update our reagents panel so that we don't miss it a second time around. And so as we progress in development, we expect this number will increase and gets completely on par with reference laboratory generated data. In this study, we also identified the presence of antibiotic resistance factors. This is extremely important. From the veterinary perspective, it's not just good enough to identify the infection, but the rise of antibiotic resistance in pathogens today is extremely frightening. And right now, they are at a loss because they end up prescribing more and more powerful antibiotics, but not always are they necessary, but sometimes they are. And so our technology is designed to be able to guide them in what they should prescribe, so we're being better stewards of antibiotic usage. This ultimately will help lessen the prevalence of antibiotic resistance if we're smart about it. Likewise, we were also able to detect dual infections, which is not equally, but important as well. We then went on to address another market. As I've mentioned earlier, food safety testing is a target market of ours. There has been multiple recalls of romaine lettuce due to contamination with E. coli 0157H7. This is one of the STEC-producing E. coli that's particularly pathogenic. And we confirmed that we could detect it off of romaine lettuce. Not only do we confirm we could detect it, we also confirmed that we can identify viable E. coli. What viable means is whether or not it's alive or dead. This is important for the industry, and we did this by performing a time course. And again, these data were generated much, much more quickly than could have been done relying on typical culture methods employed by the industry today. Down the lower left-hand corner, this gets into agricultural testing, gray mold fungus study. You may be wondering what gray mold is. I'm sure you've probably gotten a strawberry that has a gray mold on it that potentially is botrytis. Botrytis is a scientific name for it. It's a pathogen that not just affects strawberries and grapes, but is now devastating certain farms that are focused on cannabis growth. On a worldwide basis, this pathogen alone causes between $10 and $100 billion in crop loss per year. And again, this is just one of many fungi that can cause crop damage in the agricultural industry. So I certainly believe you can appreciate that farmers are looking to protect their cash crop, and they're looking to do surveillance such that they can avoid these massive crop losses. Now, these first three studies focus on pathogens. We are a genetic analysis company. We don't necessarily need to only look at pathogens. We can do much more than that. So we decided to embark on a genotyping study. And so what exactly is a genotyping study? We wanted to do something which wasn't pathogen focused, so we decided to look at the human genome. And the human genome, if you're aware, has 3 billion base pairs in it. What genotyping means is you're looking to characterize an individual for a particular gene of interest. Genes can have different variations. These variations make each and every one of us unique. Some of these variations confer either really a trait or potentially a benefit, or if it's a mutation, a defect that could be problematic. So we chose to focus on 2 genes that are of interest to the pharmaceutical industry. They are AKT1 and COMT. These 2 genes do affect the response to drugs, the risk of addiction, and side effects. We are entering an era of personalized genomics. Soon we will be at the stage where when you consume a drug, it will be dosed for you specifically, not for the general public. And the way in which we will get there is by genotyping each and every one of us such that we better understand how drugs are metabolized across us individually compared to the population. So when we did this study, we took cheek swabs and we generated 100% conclusive data. It was actually quite exciting. We had volunteers as a company, and everybody was walking around saying, oh, I'm wild type, oh, I'm mutant. But nonetheless, these are multifactorial responses, and we found it to be extremely formative, and this was just to give really the industry, I would say, an indication that our technology would be very useful for genotyping for this purpose. On to the next slide. Arguably the most important achievement to date and the most important slide in the slide deck. On Tuesday, we placed our very first beta prototype into the hands of a customer. The reason why this is a significant milestone for the company is because many companies working on diagnostics, I would say, languish in R&D mode forever and never get to the point where the technology performs to the level it needs to perform at to be competitive, and they never get to the stage where they place their technology out of their laboratory into the hands of a customer. And we've achieved that goal. We've placed it at Ethos Veterinary Health. It's actually a hospital within that organization. Specifically, it's at the Massachusetts Veterinary Referral Hospital. This is just one of 24 hospitals owned and operated by Ethos. Ethos has been a fantastic partner for us. They really are guiding us in the development of a product. This is extremely important. You want to have quality voice of customer, and right now they are a target customer for us. If we build a product that meets their needs, we anticipate them being a very happy customer in the future. I do want to mention that this is just the start of our beta program. Our beta program is intended to place beta units in potential customer sites across many different markets, continuing with veterinary diagnostics as well as food safety and other open access applications. So this will be an ongoing program. Slide number 17 and continuing to slide number 18. So the value proposition for a veterinary clinic. This slide is specific to the veterinary clinic. I do want to note that every market is different. The vet market is very different from the food safety market. Likewise, the open access market. Each needs to be treated differently. So this is specific for the vet market. And if you have additional questions, I'm happy to answer them afterwards. So first, in regards to the veterinary market, they are used to sending samples to an off-site reference lab for infectious disease testing. As such, they are not used to buying capital expense equipment for this purpose. Some labs will buy the technology right off the bat. Others will be more interested in a lease program. So we will certainly offer a lease program to veterinarians. A lease program that generates $500 a day, and some veterinarians will be higher, some veterinarians will be lower, but assuming this amount of revenue per day under a lease program, and again, we want to be an invisible slip-in solution for the veterinary practice where they aren't spending more money, but importantly, they're providing extremely high-quality care, but now in an hour rather than waiting days. And this, again, has a significant impact on the quality of care they can provide to their customers. We want them to be able to, not charge your customers anymore. Effectively, an invisible solution swap out, but again, with the benefit of time. Under this model, after just 100 days, they will have effectively paid off the cost of the instrument, and after that point, it becomes really a huge revenue-generating opportunity for veterinarians. This obviously is motivation for them to adopt the technology, and the way in which we can do this is because, again, the cost for us to generate our consumables is extremely low. Just $4 per test is the estimate at this time. And again, charging a veterinarian $100 to run a sample, which is in line with their current testing costs. These are equivalent to a nice 6% profit margin, which is extremely, extremely healthy in the industry. Now, slide number 19 details a little bit more about why we are so low cost for our consumables, because this is an important thing to understand. We have a single-use disposable cartridge. That cartridge is intended to concentrate, lyse, and purify the genetic material in the sample. After every use, it is discarded. But it is really made up of just 2 pieces of plastic, 2 filters, and a tube. Low cost to manufacture. Likewise, down below, I mentioned that this is a microfluidic instrument. It draws from a test reagent panel. This test reagent panel is placed in the instrument and stays there for months at a time. You can perform hundreds of tests before it needs to be replaced. The cost of filling these vials that make up the reagent panel is extremely low cost, and it again this form factor allows for end users who are interested in open access testing to customize that panel to detect whatever they're interested in detecting. Slide number 20 Our objectives over the next 12 months, of course, the beta program is going to be pursued aggressively. This is an opportunity for us to get our technology into the hands of potential customers. This is important for multiple reasons. One, we certainly expect them to have an excellent experience and to be excited about the technology. When they're excited, they talk to their colleagues, they go speak at conferences, they spread the word. There's no better marketing material than word-of-mouth marketing. We also need to support this beta program. This means hiring sales staff and marketing staff. This is all in the effort of building the sales funnel. And we, of course, are approaching commercialization. We need to engage a contract manufacturer. We are starting the process of lining up effectively audits to look at different contract manufacturers to select the ideal manufacturer for us. And as the beta program progresses and as we get farther along, we expect to be engaging some of these beta clients with agreements to purchase instruments. And we anticipate selling our first commercial instruments in early 2020. Slide number 21. When evaluating a company, it's not just the technology, it's the team that's bringing it to market. I'm extremely proud of the team we've assembled. Here on the top row is our board of directors, and on the bottom row is other key management staff. I do not want to forget about those in the company who are not listed on this slide. Everyone in our company is valuable and providing a key component of support for the company, and we're leveraging everybody's expertise to really guide this company in the right direction so we make the smartest decisions possible with the interest of getting to market as soon as possible and returning as healthy return of investment as possible for our shareholders. So this last slide, slide number 22, Lexagene is working on what we consider to be a very disruptive technology. It is meeting a critical unmet need in these underserved markets, which I've highlighted. To recap, a very fast turnaround time. Again, this is an instrument placed at the point of care. And if I will draw your attention to the picture, you'll see a woman here interfacing with our instrument. That's the size of the instrument. You'll see it's a very easy, attractive benchtop instrument that will fit into all laboratories. Anyone can operate it, initiate sample processing in just a matter of seconds. Again, because we are using automated microfluidic PCR, we expect fantastic sensitivity and specificity. The ability to customize, that is the open access feature of the technology, is extremely attractive, allowing us to sell into markets that currently have no solution for automated customized testing, that is. And of course, our low cost per sample test is extremely important for industries like food safety, where they're very cost sensitive. So in the end, we are building what we believe to be the first of its kind instrument for on-site pathogen, but also other molecular marker testing. So with that, I am happy and would welcome questions. So Glenn, why don't you take it over?
Thank you very much, Jack. That was great. So we've got a pile of questions in the queue. I'm going to try to combine some of them because they tend to repeat some of the same themes. So if I don't get to your particular question, just again, remember to email me, glenn@bristolr.com, and I'll get back to the email. So the first question or series of questions is around your commercialization efforts. Can you just review the path to commercialize the product, some of the costs involved, the time, and the capital needs for that process?
Yeah, certainly. So the path is first, we just started beta testing, and so let me spend a little bit more time on what beta testing means. Beta testing is an opportunity for the company to, one, start feeding the sales funnel, but also it's important for us to get feedback from our beta customers. We need to make sure we're building the right product to meet their needs, otherwise they're not going to buy the technology. So we're going to be taking to the feature requests, if you will, and ultimately locking down the design for our commercial system. So the lockdown of our commercial system will happen likely in and around the October timeframe. And so this new commercial system will, of course, be an improvement over the current beta. And it will, from our perspective, be an instrument that we have a high confidence will meet the customer's needs. So once the design is locked down in October, we then immediately start manufacturing. And the manufacturing process, again, through a contract manufacturer, does take a couple months, and we start selling in Q1 of 2020. So in regards to the capital needs to get there, obviously getting to commercial state will require additional capital. We will be hiring additional sales and marketing staff to complement what we have in-house currently. And we need to, of course, build up the inventory for the commercial systems. So collectively, this will require additional capital. We'll be raising capital as we need it. And that's basically it. You know, we expect to be needing roughly on the $7 to $8 million range to start selling instruments.
Okay, thank you for that, Jack. The next question is, would you compare this to a T2 machine or an open-gen machines, which are also presented to investors as an automated quick turn able analyzer?
So I don't like to get in the habit of commenting specifically on competitor technology. What I will say is the T2 system uses a very different way to analyze the presence of infection. It does rely on culture, and it does have a longer turnaround time, and they aren't as good, I would say, at screening for multiple pathogens at once, likewise not looking at antibiotic resistance factors. I certainly believe our technology has many advantages over the T2 platform, and for this reason I certainly welcome a side-by-side comparison to have us go head-to-head with this competitor. So as you might imagine, there are, of course, competitors out there. We welcome head-to-head comparisons in the future, and we expect our technology to perform really like a gold standard because we are building this technology to provide a very, very high-quality answer similar to that of a reference laboratory. And so these are comparisons we welcome in the future.
Okay, thank you. Do you expect the 1 hour analyzing to come down over time?
I do. We try to under-promise and over-deliver in regards to the performance of our technology. We know currently, as of right now, we can provide an answer faster than that. That said, the process of product development goes through many iterations where we continually improve the technology. And until we're on the market with our final product, we won't be publicizing a specific time to result. But at this time and point, I'm 100% confident that we can reduce that time down beneath an hour.
Okay, good. Are there any changes needed to the machine when you're analyzing pet versus food?
For pet versus food, no. A lot depends on what the food application is. On the food side, there are lots of different testing that happens. It's unfair to lump the food industry into just looking for one thing. The food industry often does a lot of swabbing of surfaces, both food contact surfaces and non-food contact surfaces. They also will process, raw ground beef and whole heads of lettuce, etc. For our technology, the way it will fit into the food safety industry is we anticipate that, one, we can provide a very quick assessment as to the risk of that food item. For items like ground beef, we have a microfluidic instrument. It does not process direct ground beef. What needs to happen is it's first placed into a broth, and then that broth is tested. The important point here is that we believe our technology can return a result in that broth extremely fast, not waiting for the 24-hour time frame that is typically required for food safety testing. So they are treated slightly different. In the animal side, you have an active infection where generally the titer of a pathogen is quite high. On the food side, it's not an active infection. It is a very, very minute quantity of pathogen, and for that reason, the food industry often does rely on culture, and we anticipate being a supplement to culture and allowing culture to return in time an answer much more quickly than currently is possible today.
Okay, thank you. Can you again address the total addressable market, and does this include the human market opportunity?
So for human market, the IVD market is roughly a $19 billion industry. And right now, we have intentionally not been making a big deal of the IVD market. The reason is because there are plenty of markets out there today that are underserved, which are great revenue generating opportunities for us because there is effectively no competition in those markets. That said, we completely expect to eventually serve the IVD market, but we want to approach that market when we are ready. And for us, what that means is we're going to succeed first in the veterinary space, food safety, other open access applications. Once we're generating a healthy revenue there, and again, this won't be soon after we're commercial, we anticipate starting the process of submitting for a 510 clearance through the FDA.
Okay, thank you. Can you describe the license for use of the microfluid technology? How long does that license last, and the bandwidth of your patent protection?
Yeah, so we've licensed 2 patents from Lawrence Livermore National Labs. The National Lab has, in many ways, an obligation to help companies commercialize technologies that were invented at the National Lab because those inventions were really the result of taxpayer supporting dollars. And so our licensing terms are very, very favorable. They're currently at 3% royalties for selling the instrument, likewise the cartridge. We also have a stacking term on our license agreement where if we are required to license additional IP to go to market, the National Lab will accept down to 1.5% royalty. This is extremely low for the industry. I also do want to comment that Lexagene is working on its own patents that will extend the protection of the company. We have one that is pending and others that are in preparation that will be submitted soon. These, of course, won't expire until 2040. The Livermore patents expire in 2028. And as a result, we have a solid period of full patent protection moving forward, both in the United States and for our pending patents, likewise worldwide.
Okay, thank you. Are there any permitting issues or hurdles that you need to overcome for the food and veterinary markets?
The veterinary market for companion animal diagnostics for instrumentation is effectively an unregulated market. For some chemistries, the USDA does regulate, but right now with our instrument, we expect that given the companion animal focus, we're not working on a drug or something that goes into an animal designed for human consumption. The USDA is much less interested, unfortunately, in companion animals. So it's a market that's very easy for us to enter with relatively small regulatory hurdles. On the food side, it is not required, but highly recommended to have AOAC certification. This is the Association of Official Analytical Chemists. It's a certification process where we compile a data set, submit for review, and demonstrate to them that our technology is equivalent, if not better, than the current standard. And we anticipate progressing on these studies. As I mentioned before, I am traveling down to D.C. to talk to the USDA and FISIS. This is the start of this process so that we have these certifications in place as soon as possible.
Okay, thank you. Can you just cover again the cost of the machine and the cartridge and then the margin to you on those products?
So the cost of the machine right now, as you can anticipate, we have just built and placed our beta prototype. We are locking down the design of our commercial system in the October timeframe, and then we're going to be forecasting sales after the beta program has progressed for a number of months. Based on the amount of our first build, you're all aware of volume manufacturing, how this can drive costs down. We'll have a much better idea of the cost of the instrument once we're farther along in October. That said, we certainly expect to be very competitive with competing technologies that are now in the human clinical diagnostic space. And, price-wise, we expect to be able to, undercut our competitors if we need to.
Okay, thank you. Next question. I'm assuming this question is referring to reference labs and to the person asking it. My apologies if I'm wrong. What percentage of testing is false positive or false negative? And then how much benefit is there to testing on site?
So that's a complicated question. So when you're looking at false positives and false negatives, you really drill down and you look at every single test the instrument performs. Our instrument, once it's commercial, negative readout. So it's impossible really to lump all of those together at this time frame because we haven't generated those data yet. Nonetheless, one of the beauties of genetics is as we go through development and we're doing this beta testing, we will continually update our tests to make sure that our false positive and false negative results is as low as possible, and we do expect it to be comparable to the quality of reference lab generated data by the time we are on the market.
Okay, thank you. Do you detect all DNA present or just the DNA from live cells?
We detect all the DNA present, and I would like to comment briefly on this. So in the food industry, the presence of DNA from, say, Salmonella is a risk factor, and it's a risk factor because with a single time point with genetics, you cannot confirm viability. The advantage of our technology is because it is so low cost to operate, we can take what's considered to be time point 0, so this is an initial snapshot of the sample, and we quantify how much DNA is present. We then can go back to that sample if it's in an enriched broth and test it, say, 4 hours later, and we've done this in the lab, and we can quantify the same genetic targets again, and if we see an amplification of the target sequences, that suggests that the pathogens were in fact multiplying during those 4 hours. And we are sensitive enough to monitor the growth of a sample over the course of 4 hours. Again, this is something which now takes the industry over 24 hours to generate a result, and we can confirm viability right now in just 4 hours for some pathogens.
Thank you. What kind of maintenance or cleaning is required for the machine?
So this is going to be to be determined. Right now, the components of the system are extremely robust. We do expect, as with all genetic equipment on the market today, generally engage customers in a PM, a preventive maintenance program, where you would have a service engineer swing by once a year to perform a series of checks and updates. So we would anticipate that would apply to us as well, where once a year we have somebody check in with the technology to make sure everything is operating appropriately. Importantly, I did not mention this in the talk, but our instrument performs a series of controls, and if these controls fail, it indicates to us that the instrument needs service. And so the instrument itself is very well controlled, and we are manufacturing it for ease of service. And so, like any instrument, there will be a PM involved, and that's standard with the industry.
Okay, thank you. Are you going to need or looking for marketing or distribution partners into the vet or food industry?
Yes, so we are a company that's been really strictly R&D up to this phase. Now that we are in beta testing mode, we are looking to expand our outreach to these customers. That includes hiring both marketing and sales staff. Right now, our staff in-house, their jobs are to advance the product in-house, not really to hit the street, so to speak, and begin selling instruments. And so over the coming months, we'll be hiring staff to support the program so that once we do lock down the first commercial build, we have a very good estimate to how many instruments we'll be selling in the first quarter.
Okay, thank you. Are there major device companies or biotechs that are aware of what you're doing, and are you having any kind of interaction with them?
There are. I mean, we are in conversations with some very big strategic biotech companies as we speak. These negotiations are ongoing. Many of these big biotech companies, monitor us over time, and they'll swoop in. Often, generally, once we are generating data, beta sites are often more likely waiting to the first commercial sales. So those conversations are ongoing, and it's an open dialogue, and we treat them as we progress in maturity and see what's in the best interest of the company moving forward.
Okay, thank you. Do you have just the one beta test planned, or do you have future beta testing planned, and have there been instances where you've talked to customers who did not want to participate in the beta test?
Well, we attend conferences, and when we attend conferences, we have individuals swinging by the booth who are very interested in our technology, and we collect their information electronically, lead generation, and we have a long list of people interested in beta testing. And so these beta instruments are going to be cycling through these prospective customers. We are stack ranking them such that we are hitting the valuable ones first, and also just being smart about it in regards to us as we begin the program. We obviously are not at the point where we're shipping beta instruments overseas, but we certainly are advancing the program as our resources allow us to advance that program. You asked me a follow-on question, which I already forgotten about. What aspect of that question did I fail to answer?
Do you have people that you've approached that have declined the beta test?
Well, I mean, not everybody's suited for our technology. Certainly not everybody in the world is going to buy our technology, and there are certain cases where customers who do genetic testing have different needs, and therefore our platform might not meet their needs. And so we don't meet everybody's needs, but there's certainly a very big niche for which we do meet their needs. And for those, there's been overwhelming support for the technology.
Okay. Will you be taking pre-orders in conjunction with the beta test programs?
We will. We don't expect this to happen until fall, but nonetheless, as we project out our sales for Q1 2020, we will be engaging beta testers, better understanding their demands, and when possible, securing a PO to help us guide our projections.
Okay, thank you. Next question: why did you choose to become a public company for your financing route?
So when we started this company, it was really just 2 patents, and the capital was available up in Canada through reverse takeover to initiate the company and start product development. And we started the ball rolling with this process. It's been good to us in the sense that we've raised the capital. We've gotten to the point where we are today, where now we're a healthy company of 21 employees, and we have a product in the hands of customers. And so it was a good path for us at the time, and we took it and we've benefited from it.
Okay, thank you. Is IDEX solely a reference lab, or do they also have a point-of-service machine offering?
So IDEX has point-of-service for blood chemistries. They do not have point-of-service for infectious disease testing. And so for this reason, this is a type of instrument that is, I would say, missing in their portfolio. Nonetheless, they are, you can view them either as a competitor of ours, of course, but they're also a potential acquirer. And so it's a group where obviously they know about us and we know about them, and we're in communications, and we're just leaving it at that. So.
Okay, thank you, Jack. We got a couple of questions in the queue remaining. I know we're tight on time now, so I'll ask you a couple more, and then I'll ask you for some closing remarks. And if I didn't get to your question, again, please just email me, and I'll sure to take care of that. Is the goal of this company to take the product to the retail market, or is the market opportunity simply too big, and you see yourself selling the product or a company at some point?
I mean, our obligation is to our shareholders. We want to do what's in the best interest for our shareholders. And in regards to whether or not we take it to the market ourselves or we allow for an acquisition to happen, this will be dependent on any term sheets that we may get. We'll evaluate each term sheet individually, given our position. Ultimately, it's a decision that goes to the shareholder vote, and we'll make recommendations. But at this point, we're positioning the company to take either path, and we'll address these decisions as they come in.
Wonderful. Thank you, Jack. There are no further questions in the queue. I'm going to ask you for any final comments, and then we'll end the call.
Comments from me? I just want to thank everybody who logged on to the webinar. Appreciate your time and your interest in the company. And if you do have follow-up questions, feel free to have Glenn contact me, and we'll answer them to the best of our ability.
Super. Thank you, Jack. Thank you to our audience. This now concludes the webinar.
Thank you.
Thank you. That does conclude today's webinar and telecast. You may disconnect your line at this time and have a wonderful day. We thank you for your participation today.